So in this lab, we are going to look at cell physiology. And to do that, we are going to use what's called a spectrophotometer. And I'll explain more about that in just a moment. So the main part of the cell that we're looking at today is going to be the cell membrane. And the cell membrane is a fluid mosaic model. And what that means is that the membrane is a fluid structure and it has a mosaic of proteins embedded and attached to it. Now, when we look at the cell membrane, the function of the membrane is to regulate what goes into and out of the cell. It's what we call selectively permeable. Some things can cross the membranes and others cannot. In terms of the components of the cell membrane is made of primarily phospholipids, and phospholipids have a hydrophilic head. That's what you're seeing in this diagram on the right, the orange little circles. Those are the hydrophilic heads. And in the middle of the membrane, we have the hydrophobic tails. Hydrophobic refers to water-fearing. That part is the fatty acids, and they are repelled from water. And so those will orient themselves so that the hydrophobic tails are in the middle, and the hydrophilic heads face both outside the cell and inside the cell. Now, the cell is not only phospholipids in its membrane, it also has proteins in it. And those proteins can serve a variety of purposes. They can act as protein channels. So you can see this pinkish purple one here. That's a protein channel that allows things that normally can't cross the membrane, like ions, for example. It allows those ions to cross the membrane. We can have proteins that act as enzymes in the membrane. We have proteins that can act for adherence to adhere cells to either other cells or to surfaces. We have proteins that will play a role in signal transduction, basically meaning how cells communicate with one another. And so if we look in a cell membrane, a red blood cell, for example, has 50 different types of proteins embedded in its membrane. In addition to phospholipids and proteins, we also have cholesterol in the membrane. And when we think of cholesterol, usually we think of a negative connotation, meaning we usually think of cholesterol as being bad. But in fact, your cells do require cholesterol in the membrane because the cholesterol serves the function of maintaining the correct fluidity of the membrane, meaning that it prevents the phospholipids from packing in too tightly, which would make the membrane be too solid where nothing can go in and out. And it also prevents phospholipids from spreading out too much. It acts as a patching substance, because if the phospholipids spread out too much and the membrane becomes too fluid, well, then now it's not selectively permeable and it's not regulating what goes in and out. That means anything can come in and out. And so we also have cholesterol in the membrane as well. So cells can survive in environments at a particular pH. And this is different for each type of species and even for different cells within an individual. For example, the cells in your stomach are used to an acidic pH and can survive at a pH of 2. Whereas in your intestine is much more alkaline. It's a pH of about 8. Those cells have a different pH that they survive in. And so even within one organism, you can have cells that survive at different pHs. If a cell is exposed to a pH outside of its optimal range, the membrane will be negatively affected, meaning that this could cause the cell to die. The reason is if you think about what the cell membrane is made of, remember that I said that one of the components of the cell membrane is going to be the proteins. Proteins are three-dimensional structures, and if you remember back to our macromolecule lecture, proteins only function if they're in their correct conformation, meaning if they're folded properly. If the environment of that protein changes, for example, temperature or even pH, like we're looking at today, pH, hydrogen ions, are going to influence the way that protein folding occurs. Because if you have extra hydrogens in the solution, the protein in the membrane might start interacting with those hydrogens rather than interacting with itself. And it might cause the protein to denature, which means unfold. And so you can imagine if you have this globular protein in that membrane and it denatures and it unfolds, essentially what you're going to end up with is a big hole in your membrane. And as a result, the cell is going to die. And so cells have to be within their correct pH in order for the cell to survive. If not, the membrane will be negatively affected and the cell will die as a result. So if we look at different crops, those cells will survive at different pH ranges. For example, potatoes will survive in soil at a pH between 4.8 and 6.5. Carrots a little more alkaline towards neutral, so 5.5 to 7. Onions 5.8 to 7. Lettuce has a fairly narrow range between 6 and 7. Tomatoes 5.5 to 7.5, and cabbage 6.0 to 7.5. So what you'll notice is that each of these crops has a different range of pHs that the soil can be that allow the plant to grow. And so what you're doing in this lab today is that a farmer comes to you and they want to know what is the optimal pH to grow beets. And so the first part of the lab is going to investigate what would be the optimal pH to grow beets. So on the top left, we have our beets, right? And so beets have this red pigment, and that is because they contain a type of pigment called beta-cyanin. And beta-cyanin gives off that red color. And so this is on the right, this bottom part, this is cut up beets. But what we're looking for is normally the beta-cyanin pigment would be inside the cell. And we want to see if the cell membrane gets damaged, what is the pH that's going to prevent that from happening. So if we have our beet cell, here's my cell, and inside my beet cell, I have my beta-cyanin. So the beta-cyanin pigment is going to be within the beet cell. Now, as long as the membrane is intact, that pigment is going to stay in the cell. However, if the cell membrane gets damaged, what's going to happen is that the beta-cyanin is going to come out of the cell if the membrane becomes damaged, and it's going to leak into the surrounding solution. And so what we're going to be doing in this experiment is putting beet cores, pieces of beet, in various pH solutions. And we want to see which pH solution causes the least amount of damage to the beet cell. And essentially what we're looking for is which one is going to keep the pigment inside the cell, meaning it doesn't end up in the solution. So here's a video demonstrating our setup for this. So in this experiment, we're going to look at the effect of pH on the membrane for a beet. So what I have here is I have my 5 beet cores. Each of them were cut to 3 centimeters in length. And so when I go to cut these, the mechanical action of cutting these actually damages the beet cells. So I need to start by rinsing them first. So I'm going to take my beet cores and I'm going to place them in this netting. And I'm just going to pour some tap water over them just to rinse off any pigment that is just from damaging due to the cutting. So I have my beet cores, and over here I have various pH solutions. So we have our pH 3, our pH 5, 7, 9, and 11. And so what I've done is I've taken a test tube, and for each of the test tubes, I've added 5 milliliters of the solution into that test tube. And so now I'm going to take my beet core, and I'm going to add one to each test tube. So I'm going to add one to the pH 3, pH 5, 7, 9, and 11. And I'm going to let those sit for 5 minutes. So our 5-minute time has elapsed. So now what I'm going to do is I'm going to dump off the pH solution into the waste beaker. So I'm just going to get rid of the pH solution, being careful not to lose the beet in the process. Okay. So I'm going to do that for all of my tubes. Alright, so now that I've dumped off the pH solutions, I'm going to add 10 milliliters of deionized water to each of these tubes. So it's just going to be simply one pump. And I'm going to let those sit for 30 minutes. And while that 30-minute incubation is going, we are going to learn to calibrate our spectrophotometer. So what we have here is our spectrophotometer. And so we already have it warmed up, and so this knob here on the front left, we just turned it to get the spectrophotometer on. And so this has been warming up for about 20 minutes now, and so we need to calibrate it with nothing in the machine. So here is our sample compartment, and this is where we will put our cuvettes when we go to read this out. But we need to start by calibrating the machine with nothing in it. So I always check the sample compartment to make sure that there's nothing in it, and then close the lid. You want to start with the machine set to transmittance. If for some reason it was not set to transmittance, you would just hit this mode button until you were at transmittance. Now, what I need to do is my wavelength. When I do this experiment, my wavelength is going to be set at 480. So this top knob here, we want to adjust this to read 480 nanometers. That's the wavelength of light that's going to pass through our sample. We need to blank the machine with nothing in it, which means that, notice we have a number on here. So for transmittance, we need to make it read 0. So to blank transmittance, we use the knob on the left. And so we're going to adjust. It was too far. Okay, there we go. So now it's reading 0 on transmittance. So now we're ready to blank with our sample blank. So when we do this, we are no longer going to adjust this knob on the left. That is only when nothing is in the sample compartment. So at this point, you're not going to touch this knob on the left. We will only work with the one on the right. The other thing is down here on the bottom, there is a filter level. And so on this side, it's 340 to 599. On this side, it's 600 to 950. We are using a wavelength of 480, so the lever is over here on the left. So notice there's a little switch here. I need it to be on 480. So this is our cuvette. It's basically a smaller test tube. It has a special kind of glass that will allow light to pass through it. And we need to be careful when we work with our cuvettes that we don't touch them on the bottom. The reason for that is that the oils on your fingers will make marks on the test tube or on the cuvette, and the light won't pass through. So to start, before we put the cuvettes in, we're going to take some lens paper, and we're going to wipe off any fingerprints that might be on the cuvette, because you have to think about that you don't know who touched this before you did. So I wipe off any fingerprints, and then I'm going to open my sample compartment, and you'll notice that there's a white line on the cuvette. There's also a line on the spec, and so I need to make sure that those white lines match. Let me re-wipe that. So I'm going to line up those lines, and I'm going to close the sample compartment. And so now I need to blank the machine. In that cuvette is the water that the beads are placed in. And so it's the same solution that the beads are going to be in. So now I want to go to mode, and I want to go to absorbance. And you'll notice that the machine is flashing 1999. I need to tell the machine that we're basically, think of it like tearing the scale, right? When we are weighing something on the scale, we need to zero out the scale before we begin. Same thing when we use our spectrophotometer. We need to zero out or blank out the solution that the beads are in, so that is showing the machine that that represents no pigment. So again, we're not going to touch the knob on the left, but we are going to use the one on the right, and we need to make absorbance read 0. So sometimes you have to play with it to see which direction. All right, so we need to make it read an absorbance of 0. So this is telling the machine that there's no pigment in this solution. Now they can be a little bit finicky. There we go. So it was reading an absorbance of 0. If it's off by 1/1000th, it's fine. So I'm just going to now take my blank out. So this is my water. I'm going to take it out and I'm going to put it aside. And now my spectrophotometer is ready to put my sample cuvette in. So our test tubes with our beats have been going for 30 minutes. And so now what I'm going to do is I'm going to take the beat and the solution and I'm going to vortex for 5 seconds. And the purpose of that is just to mix any pigment that's settled in the tube. Okay, so now I'm going to take my test tube, and what I'm going to do is I'm actually going to temporarily put the label on the top, just so I can keep track of which one is which. And then I'm going to pour into the cuvette. You want to make sure that your beat doesn't fall out. And so there is my solution. So that's my 3. And I'm going to repeat that for all of these. And now we're ready to use our spectrophotometer. So again, in the beat experiment, you have your beat cell, and in the beat cell you have the beta-cyanin. If the membrane is intact, the beta-cyanin will stay in the cell and will not go into the solution. However, if that pH affects the membrane negatively and it damages that cell membrane, that is going to cause the beta-cyanin to come out of the beat cell and go into the solution. And so what we're doing is when we expose these beats to different pHs, we're seeing essentially under which condition is the pigment the most red. Because the more red the pigment, the more damage there was to the beat cell. And so we need to talk about how do we determine how red the solution is. So the way that a spectrophotometer works is that we have a light source. So here's our light source here. And we have this diffraction grating. And this diffraction grating allows only certain wavelengths to pass through the machine. And so in our experiment, we set our spectrophotometer to 480 nanometers of light. And so only that wavelength can pass through. So the light is going to go through the sample and it's going to be measured by a detector. When we start and we blank the machine, we're using what we call a blank sample. And what that basically is that is the same solution that your sample is in. In our experiment, our blank is going to be the deionized water because that's what our beats are ultimately in when we put that sample in the spectrophotometer. So our cuvette has just water. And what we're basically doing when we're putting a blank in is that we're telling the machine that that amount of light that's passing through represents no pigment. Essentially, you can think of it like when you tear a scale. Like if I wanted to cancel out the weight of the beaker before I measured water, I could put a beaker on the scale, push tear, it's going to subtract out the weight of the beaker so that I'm only measuring the water specifically. Same idea here. In this case, we're using a blank cuvette, and in that, we're basically telling the machine that that amount of light that passes through the sample represents no pigment because we know there's no pigment in that deionized water. Now, what happens when we put our experimental sample in? Well, when the experimental sample is placed in the cuvette, any particles that absorb light at that wavelength that you set will read as being absorbed. So if I have two solutions, let's say I have solution A and I have solution B. And so the solution in A has less pigment than in B. So what happens is when I pass light through it, the light is going to pass through because there's less particles. And so what ends up happening is when we look at the detector, so we'll just say this is the detector. For the solution with less pigments, so decrease pigment, we have an increase in transmittance, meaning more light is able to pass through, which means we have less light absorbed. So if we have a decrease in pigment, right, the lower the pigment, the lower the absorption. On the flip side, in solution B, if I have solution B and the light is going to pass through the sample, if that pigment absorbs that wavelength of light, right, if it absorbs that wavelength of light, less light is going to pass through. So some of that light is being absorbed by those pigments. And so now in this tube, we have a higher amount of pigments. What that means is we get a lower transmittance because less light is going through, which also means we have a higher absorbance. So notice that the more particles in the sample, the higher the absorbance. So that would be like in solution B, right? The more there's pigment, the higher the absorbance. So if we think about it in our beet experiment, are we looking for the solution that has the lowest absorbance or the highest absorbance? Remember, we are trying to figure out which pH to grow beets at. What that means is we're looking at which pH is optimal. The pH that's optimal, is that going to cause more damage or less damage? The optimal pH is going to have the least amount of damage, right? The optimal pH has the least amount of damage, which means that the pigment is going to stay in the beet and is not going to be in the solution. So in this case, the least damage has the lowest pigment. So the optimal pH is going to have the least amount of damage, and that cell is not damaged, and therefore the pigment stays in. And so if we have a low number of pigments, that means we also have a low absorbance. So we are using our spectrophotometer essentially to measure how red the solution is. Basically, how much pigment was being released from the beet cell. Because the more pigment that's released, what that tells you is that pH damages the membrane the most, and that is why the pigment came out and into the solution, and therefore those pigments are going to be absorbed and you get a higher absorbance. So in our beet experiment, we are looking for the optimal pH, which means it has the least amount of damage, the lowest amount of pigment in the solution, and the lowest absorbance. So the one with the lowest absorbance is going to be the optimal pH to grow beets. So our spectrophotometer was already blanked, so now we just need to read our samples. So we're going to start with pH 3. I'm taking my cuvette and I'm wiping off any fingerprints, again, so that the oils from my fingers do not affect it. The line is going to match up with the line on the cuvette holder. So all I do is I put the cuvette in, I close it, and I record the absorbance that's on our spectrophotometer. So I take that one out. Now I'm going to go to pH 5. I'm going to wipe off any fingerprints that might be on this cuvette. I'm going to simply put it in the cuvette holder, and I'm going to record the absorbance for pH 5. I'm going to take out 5. I'm going to move on to pH 7. Put it in, close the sample compartment, record the absorbance for pH 7. pH 9, wipe the fingerprints off, put it in the sample compartment, close it, record the absorbance for pH 9. And finally, pH 11, wipe off the fingerprints, put it in the sample compartment holder, and record the absorbance for pH 11. And so now you have your data for the beet experiment. So just to summarize what we did in part 1, we took our beet cores, and we had 5 of them, and we placed them in various pH solutions and let them sit for 5 minutes. And when we did this, we did a pH of 3, 5, 7, and 9. After the 5 minutes is up, we dumped off that pH solution, and we soaked the beets in deionized water. and we let those sit for 30 minutes. And so what ends up happening is any damage that was caused by the change in pH would then cause the pigment, the beta-cyanin, to leak out of the cell, and the pigment would become more red. So we mixed our beet and our deionized water. We poured the water, just the water, not the beet, into the cuvette, which is our test tube for the spectrophotometer. And we measured the absorbance of each beet water using the spectrophotometer. And so these are our actual results. So you could see the colors that each solution was before we put it in the spectrophotometer. And you can already see just by looking which pH is not optimal. You can see which ones cause the most damage just visually. In terms of which one is best, it's a little more difficult to determine which one is the least red. So again, this is why we use our spectrophotometer. It gives us an idea of which one actually is the lowest absorbance. So this is just to summarize your data for table 3-1. So what I did was I took pictures of the spectrophotometer and I indicated the pH next to it. So you have your data. So you're going to record your absorbance, and the absorbance, for example, for pH 3, you're going to record 1.270. That's your absorbance for pH 3. Record it for 5, 7, and 9, and 11 in table 3-1. You are going to delete table 3-2 because obviously we don't have class data since you're not in class. You're just going to, when you graph, you'll graph table 3-1 instead. So the next experiment is going to be looking at the effective surface area to volume ratio on the rate of diffusion. Now, why is diffusion important? So if you think about your respiratory tract, for example, right? When we breathe in, we take in oxygen. The oxygen has to go from our lungs and into the blood. And the blood is going to then circulate, and it's going to drop off oxygen at all the tissues in your body because your body needs the oxygen. Those cells need the oxygen to do cellular respiration and to make ATP. And so we need to have adequate diffusion between the blood and the cells, for example. That's going to allow for absorption of oxygen into the tissues. So when the blood comes in, it's going to deliver oxygen to the tissues. One of the things you'll learn later on is one of the places where gas exchange occurs is going to occur at the capillaries. So your capillaries are very small blood vessels. They slow flow of blood through those blood vessels so that we can get gas exchange between the blood and the tissue. So this is just showing you, if you look at your blood vessels, O2 needs to come from the blood and it needs to go into the cell. Same thing with glucose, for example, right? You need to get glucose to the cells to be able to do cellular respiration. You also need to expel CO2, carbon dioxide, as well as other waste products. And so you need to have this diffusion to occur in the tissue. So in this experiment, what we're looking at is the relationship of surface area to volume. So you can think of this surface area analogous to the amount of cell membrane, because remember that we're looking at the cell membrane in this experiment. So when we do this, we're going to be using potato cubes, and the potato cubes are going to represent a cell. So you're going to have a cube of a particular size. The surface area is the area on the outside. So again, think of that like the amount of membrane. The way to calculate the surface area is you're going to take the length times the width times the number of sides. So if it's a cube, there will be 6 sides times the number of cubes. So if I have a cube and my cube is 2 centimeters on each side, my length would be 2, my width would be 2. So 2 times 2 times 6 sides times 1 cube. My surface area would be equal to 24 centimeters squared. Don't forget units. You need to write centimeters squared. That's because of the length times the width, right? Because length would be centimeters, width would be centimeters. So my units, centimeters squared. The volume is equal to the content inside the cell, right? The amount of space that the cell occupies. So to calculate the volume, you're going to take the length times the width times the height, because you're taking the whole volume inside. So length times width times height times number of cube. So if I have a cube that is 2 centimeters, I'm going to calculate the volume as 2 times 2 times 2 times 1 cube is 8 centimeters cubed. My surface area to volume ratio, right, the volume relative to the surface area. So if I take the surface area and divide by the volume, my ratio would be 3 to 1. My surface area is 3 times larger relative to my volume. Look at this example of various size cubes. So if we had a 2-inch cube on every side and we calculated the surface area, it would be 24 square inches. Volume would be cubic inches. So make sure when you're indicating your volume, you're putting cubed. You are going to have a surface area to volume ratio of 3 to 1. If I have a cube that is 4 inches on each side, the surface area would be equal to 96. The volume would be 64. The surface area to volume ratio is 1.5. If I have a cube that is 6, notice my surface area to volume is 1. If I look at the 8-inch cube, it's 0.7. So notice that for this, the larger the cube, the lower the surface area to volume ratio. So the bigger the cube, the lower the surface area to volume ratio. The smaller the cube, the higher the surface area to volume ratio, right? Because if you think of a small cube, it has less volume relative to its surface area. Its surface area is actually very high. So the smaller the cube, the higher the surface area to volume ratio. Notice that these are inversely related. When one goes up, the other goes down. So the bigger the cube size, the lower the surface area to volume ratio, and the smaller the cube, the higher the surface area to volume ratio. So on page 40, it has some calculations for you to do to compare one 3x3 cube versus 27 1x1 cubes. And so let's take a look. So we're looking at what effect will cutting a 3 centimeter cube into 27 1 centimeter cubes have on the rate of diffusion. So if we take that one big cube and instead we cut it into 27 smaller cubes, right? The bigger the cube, right? The higher the cube is, the smaller the surface area to volume ratio. The smaller the cube, the higher the surface area to volume ratio. So what we're trying to see is which one will have the faster rate of diffusion. Will water diffuse faster in the small cube or the large cube? And so you can think of this like, for example, the purple represents something that's in the cell. If you're looking at a big cube, is it going to take more time or less time for that to get out of the big cube versus the small cube? And so it's kind of like that. But in this case, we're looking at which one has the fastest rate of diffusion by measuring the water that the cube will absorb. So let's take a look at it. So what you would be doing in this experiment is you would cut potatoes to the appropriate size. So again, you would cut one cube that is 3x3x3. You would also cut 27 small cubes, and each cube would be 1x1x1. Overall, they are the same size. It's just that the other cube is cut into smaller pieces. So what you'll do is you would take your cubes that you've cut, your potato that you cut, and you would record the weight of the large cube and the small cubes. So you would record the weight before. Then you would submerge the potatoes in water and let them sit for 45 minutes, making sure that the potatoes are completely submerged. After the 45 minutes, you would take the potatoes out, lightly pat them just to get rid of any extra water that's on the outside, and then you would re-weigh the potatoes. And what you're looking for is which ones gained the most weight. Did the large cube gain the most weight, or did the small cube gain the most weight? So you will determine weight change as well as percent weight change, and then you will graph the data for the percent weight change. So for this one, you'll fill in the data on table 3-3. And so 3-3, you'll notice we have 27 1 by 1 by 1 cubes. You're going to record the initial weight, and I'm going to show you the experiment in just a minute. You are also going to record the initial weight for the large potato cube. You can cross out table 3-4 because, again, that's class data. You won't have class data in this experiment. You will only have the individual data. And so let me show you how this experiment would be done. So our potato cutter just cut our pieces. And for this particular cutter, we are looking for potato cubes that are 3 centimeters by 3 centimeters by 3 centimeters. So you'll notice that there's a lot of these scrap pieces. We don't need that for this part. We just need the bigger cube. And so I'm going to take this, and it already cut it 3 centimeters by 3 centimeters, but I need to make the length 3 centimeters as well. I want to be sure not to include any of the skin in the piece that I use. So I'm going to start by cutting off an end first. Nope, that still has it. Okay, so I've made my end, and now I'm going to use my ruler, and I'm going to measure 3 centimeters. So let me see where that is. There we go. And so now I have my cube that's 3 by 3 by 3. We also did this with another potato cutter, and the other potato cutter was used to make 1 centimeter by 1 centimeter by 1 centimeter cubes. And so what we have in this dish is that we have 27 of those 1 centimeter cubes. So now what we need to do is we need to record the weight of the potatoes. So we'll start with the 3 by 3 potato first. So I have put my weigh boat on my scale, and I push the tare button to subtract out the weight of the weigh boat. And so now I'm going to put my potato on here, and I'm going to record the weight of the potato before. So you can record the weight of the large potato in table 3-3. This is the weight before soaking in water. So we recorded the weight of our 1 3 by 3 cube. So I'm going to take that one off, and I'm going to make sure that my scale is still tared at 0. And now I'm going to add my 27 small cubes. So I've added them, and now we're going to record the weight. Now you can record the weight of the small cubes before soaking in water, and this will go in table 3-3. Okay, so we've recorded the weight of both sets of cubes, and now we're going to take those cubes and put them into a beaker. So the 1 large cube goes in one beaker, and the 27 small cubes go in another beaker. And so what I'm now going to do is I'm going to submerge them in tap water. So this is just some tap water. And I just need enough tap water so that the potatoes are fully submerged. So now that my potatoes are submerged, I can let them incubate for the appropriate time. And then after that time, we will record the new weight of the potatoes after they've been sitting in the water. So we are going to look at the effect of surface area to volume ratio on the rate of diffusion. So our potatoes have been sitting in our tap water for 45 minutes. And remember that we recorded the weight before the potato was in the water. Now we're going to record the weight after the potato's been in water. So what I'm going to do is I'm going to dump off the water. Okay, so I have my potato, and I'm going to gently pat it just to get rid of any excess water that's on the outside. You don't want to pat too much or it's going to wick the water out. You just want to lightly pat it. My scale needs to be tared, so I'm going to push tare. And now I'm going to take my potato and I'm going to place it on the scale, and then we're going to record the weight. So you are going to record the after weight for the large cube. And so on the right, we have after soaking in water, and you can see on the scale the weight after. And so you want to record that in table 3-3. The same procedure was repeated with the 27 1x1x1 cubes. So I patted them off, I reweighed them, and you can see on the next slide the weight of the small potatoes after. So here we have our weight of our small potatoes after soaking in water. And so what you're going to do is you're going to go back now using this data, using the before and after weight of the water. You'll notice that, for example, if you look at the small cubes, that they gained weight, right? The weight went up. That makes sense because we're expecting that water would diffuse into the potato cubes. So we do see that it gained weight, so that is good. But you need to calculate the weight change and then the percent weight change. And then you're going to compare that to see if it was the large cube or the small cube that had the greatest percent weight change. And that's going to give you an idea of the effect of surface area to volume ratio on the rate of diffusion. We have another experiment looking at the effect of surface area to volume ratio on the rate of diffusion. And this one is a little more visual. It allows you to see it by color change rather than weight. And so what we're doing in this experiment is we're cutting two cubes. One is 3x3x3, one is 1x1x1. And we're taking these cubes and we're putting them in vinegar and we're letting them sit for 15 minutes. As the red cubes absorb the vinegar, they will turn yellow. And so we will look and see how much vinegar diffuses into each cube and then use that to answer summary question number 5. And that will be explained in the next video. So in this experiment, we are looking at the effect of surface area to volume, just like in the potato experiment. So what we have is we have these phenyl red agar cubes. And phenyl red is a pH indicator. And phenyl red, when alkaline or basic, is this magenta pink color. Phenyl red, when neutral, is orange. And when acidic, is yellow. So what I have here is I have these two cubes. I have a large cube that's 3x3x3. And I have a small cube that's 1x1x1. And so I have both of my cubes in my dish. And I'm going to submerge them in a vinegar solution. So if you think about the pH of vinegar, vinegar is going to be an acidic solution. And so watch what happens to the cubes when they're submerged in vinegar. So I'm just pouring enough to cover the cubes. So both of my cubes are submerged, and I'm going to leave them in the vinegar solution for 10 minutes. And after 10 minutes, I'm going to look at the effect of the rate of diffusion based on surface area to volume ratio. So our phenyl red cubes have been sitting in the vinegar for 10 minutes, and let's take them out and take a look at what those cubes look like. So this is the larger cube, and notice that the larger cube, this was our 3x3 centimeter cube, and you can see that the vinegar diffused in, but not all the way through. If you look closely, you can see that the middle of the cube is still pink. But look what happened to our small cube. So this is our 1x1x1 cube, and you can see that the vinegar has diffused almost completely through. And so this experiment is looking at the effect of surface area to volume ratio on the rate of diffusion. So here are the results from the agar cube experiment. So notice that on top we have the large cube, and you can see that for the large cube that the vinegar did not diffuse all the way in. However, with the small cube, the vinegar diffused almost completely. And then on the right, what we did was we cut it in half so it was easier to see how much it diffused. And clearly you can see that the vinegar diffused better into the small cube than it did with the large cube. And so you want to think about how this is affected by the surface area to volume ratio. What does that tell you about the surface area to volumes ratio on the rate of diffusion? Is it the larger the surface area to volume ratio or the smaller the surface area to volume ratio that has the faster rate of diffusion, and why? So this is just to kind of further elaborate on why surface area to volume ratio is so important. So if you look at your lungs, you have these tiny little sacs in the bottom that are referred to as alveoli. And this is where the gas exchange is going to happen primarily. And the alveoli have these little sacs because that increases the surface area to volume ratio. By having those little sacs, there's a lot more surface area relative to their volume, which is going to help with diffusion of gases. So I just wanted to give you a little bit of information about how to go about answering your summary questions. So number 1 says, what is the correlation between the level of the spectrophotometer's absorbance readings and the amount of damage to the beat cell? So the first one says low absorbance. What you want to write there is, does the low absorbance mean that there is less pigment in the solution and less damage, or more pigment in the solution and more damage? So you want to relate the absorbance to not only the damage, but the amount of pigment. And so make sure you include that in both your answer for the low absorbance and the high absorbance. For number 2, you're going to cross out table 3-2. We didn't do the class average. Instead, this is going to be based on your answer from table 3-1. And I'll talk about the graph in a minute, but basically it's asking, what is the optimal pH for beets? So in your answer, you need to explain, use your data. The optimal pH to grow beets is blank because it had an absorbance of blank. This indicated that the beet cell was damaged the least because the least amount of pigment was released, etc. Whatever your answer is going to be. But you want to explain with your data. Be thorough. Question number 3. Based on the class average, again, you're not using the class average, so you're going to do this with table 3-3. Your calculations of surface area to volume ratio in the graph that you constructed. How does the surface area to volume ratio affect the diffusion efficiency between the small and the large cubes? So in this one, you want to answer, is it the smaller cube or the larger cube that diffused faster? Did that cube have the higher or the lower surface area to volume ratio? And then you want to explain with your data. So this potato cube, whichever one you're going to say, I'm not going to tell you which one, this potato cube, either large potato cube or small potato cube, this potato cube diffused the fastest because it had the highest percent weight change of blank compared to potato cube B, whichever one you're putting in there, which had a percent weight change of this. This indicates that, is it higher or lower surface area to volume ratio causes the faster rate of diffusion. So again, you want to be very thorough and you want to support your answer using your data. So for number 4, based on your class average data, you're not using your class average data, you're using your own data. Was your prediction supported? Why or why not? So you were supposed to predict if you thought which surface area to volume ratio would have the faster rate of diffusion. So hopefully you read through your lab and you saw that. And then you're going to answer what your prediction, if your prediction was supported, why or why not. So if you said, for example, the lower the surface area to volume ratio, the faster the rate of diffusion, well, then you're going to answer that. You're going to address that in this question, that your answer was, or your prediction was supported, or your prediction was falsified. Number 5, based on your observations and measurements from experiment 2B, this is the vinegar example. If each cube represented a living cell and vinegar represented a substance needed for the cell to survive, such as sugar, what problems might exist for the larger cell? So again, you want to explain in surface area to volume ratio as well. So did the large cube have the lower surface area to volume ratio or the higher? And then what problem might that have? Question number 6. If you compared the body heat loss of 2 different Arctic animals, a large polar bear and a small snowshoe hare, which would lose all their body heat the fastest? And it says, explain your answer with respect to differences in the overall surface area to volume ratio between the 2 animals. Again, it comes back to higher or lower surface area to volume ratio has the faster rate of diffusion. So you need to look at your data and see which one has the faster rate of diffusion. If, let's, for example, you said lower, this is not necessarily the answer, I'm just saying. If you said it was the lower surface area to volume ratio, you also want to indicate was the lower surface area to volume ratio for the large animal, the polar bear, or the small snowshoe hare. So think of it that way. Which one will lose heat the fastest? Meaning their heat will diffuse out of their body faster? Would it be the higher the surface area to volume ratio or the lower the surface area to volume ratio? And so that's what you want to do for question number 6. Now for graphs, you have 2 graphs for this experiment. You have one looking at the effect of pH on the beat cell membrane. And so what you're going to be graphing for this is the absorbance and the pH of the solution. Now remember that your x-axis, the one going horizontally, that is going to be your independent variable. Your y-axis, or going vertically, is going to be your dependent variable. So you want to ask yourself which one depends on which one. So does the absorbance depend on the pH of the solution? Does the pH of the solution depend on the absorbance? So which one makes sense? So if you said that the absorbance depends on the pH, that's the correct one, right? Because the absorbance, the amount of pigment released, will depend on the pH. So I'm going to have the absorbance on my y-axis, and I'm going to have my pH of my solution on the x-axis. Now, we did this with various pHs. We did this with pH 3, 5, 7, 9, and 11. So you're going to put those on your graph. Now, you're going to use the absorbances, use your scale depending on your data. So look at your data, look at what your highest number is, and then make sure to appropriately distance your scale. Now, I'm just going to make data up just because I just want to show you what this could look like. For example, if this is 1 and this is 2 and this is 3. So again, I'm just making this up just because I want to show you. You have 5 solutions, 3, 5, 7, 9, and 11. What type of graph should you use for this data? Answer is you should use a line graph. This is continuous data. You can extrapolate that pH 4, which is in between 3 and 5, would be somewhere in between. So I'm just going to pretend to make up data. This is by no means your data at all. Okay, so you're going to have your data points based on your table. And then using a ruler, you want to connect your dots. Don't mind the little squiggly. So you're going to connect your dots. Now, this is what your graph would look like. You also need a title. I'm going to give you the title for this one to give you an idea. You want to be thorough. So this title seems a little long, but I want to be thorough. So my title says, Determining the Optimal pH to Grow Beets Using a Spectrophotometer Set at 480 nanometers and Exposing Beets to Various pH Solutions. So I'm giving you the title for this one. That's going to be your title. And so you want to look at your graph and you want to determine what is the optimal pH to grow beets. So again, are we looking for the lower absorbance or the higher absorbance? So think about what that means. If we have a low absorbance, do we have less pigments or more pigments? Remember we said if we have a low absorbance means that we have less pigment, right? Not as much pigment is being absorbed. So lower absorbance equals less pigment, which means it has the least amount of damage to the cell membrane. So you'll look at your graph and you'll use your graph to determine what is the optimal pH to grow beets. Your second experiment was looking at the effect of surface area to volume ratio on the rate of diffusion. And so what we did is we were looking for percent weight change and potato cube size. So we need to ask ourselves, which one depends on which? So we can say either percent weight change depends on potato size, or potato size depends on percent weight change. And what you'll notice is it's the first one, right? My potato cube size can't change depending on my percent weight change. But the percent weight change can depend on the potato size. So I'm going to have my percent weight change on my y-axis. My x-axis, I'm going to have my potato cube size. Make sure that you put your units. So in this case, our potato cube size were measured in centimeters. And then I'm going to put that I have two samples in this one. I have my 27 1x1 cubes, that's in centimeters, or I have my 1 3x3 cube. So in this example, is this one going to be a bar graph or a line graph? Answer is this is discrete data, so this is going to be a bar graph. Again, I'm not putting data on here. I'm just going to, you need to fill in your scale, but I'm just going to make up random data. So you're going to have one bar for that, and you're going to have another bar for this, and then you'll shade those in. And then you also need to give it a title. Again, I'm going to give you the title for this one, but starting on the next lab, you will start coming up with the titles yourself. So your title for this one, determining the effect of surface area to volume ratio on diffusion rates. On diffusion rates using different size potato cubes, right? That's what we're trying to accomplish in this experiment. What is the effect of surface area to volume ratio on diffusion rates using different size potato cubes? That's your title. So you're going to have two graphs that you have to make, one for the pH experiment and one for the surface area to volume ratio experiment. And so lastly, we have our quiz 3 study guide. Know that all cells are surrounded by a cell membrane. Know that the spectrophotometer is used to measure the absorbance of light as it passes through the solution. You want to understand the relationship between cell damage, the amount of pigment in the solution, and the absorbance on the spectrophotometer. Meaning, is it the lower the absorbance, the least amount of damage, and the least amount of pigment? How are those related? So that's for that one. Know how pH solutions outside of a cell's optimal pH range affects the membrane. What happens to the cell membrane when the pH is outside of its optimal range? You do not have to memorize your data. You don't have to memorize which pH was the optimal pH. But if given a graph, you should be able to look at the graph and look for and identify which would be the least damage, meaning what would be the optimal pH. So that's the other one, be able to determine the optimal pH from a graph, understand the relationship between surface area to volume ratio and how this affects the rate of diffusion. So the smaller the cube, the higher the surface area to volume ratio, and does that increase the rate of diffusion or decrease the rate of diffusion? So that's what that's asking. And then, do large animals or small animals lose heat faster and why? So that goes back to that last summary question. So you want to make sure that you understand that. Read lab number 4, which is going to be photosynthesis, and define the terms that are left blank. And so that concludes lab 3.